Bismillahirrahmanirrahim. New topic of our discussion is Krebs cycle, which is sometimes also called as the TCA cycle. Krebs cycle को citric acid cycle या tricarboxylic acid cycle भी कहते. That's why this abbreviation TCA is actually put forth. Or Krebs cycle mainly happens in the mitochondria of all the cell because this metabolic pathway is one of the key player of the cellular respiration. That's why it is also called as the mitochondria is called as the energy house of the cell because uh, दो metabolic processes जो energy production में mainly involved हैं वो चूँकि mitochondria के अंदर होते हैं. That's why mitochondria is called as the powerhouse of the cell. One of that metabolic pathway is the Krebs cycle and the other one is the electron transport chain. So एनर्जी प्रोडक्शन में सिट्रिक साइकिल का या टीसीए साइकिल का या क्रेब्स साइकिल का सेंट्रल रोल है because all the different metabolites, the intermediates, they ultimately they join basically the citric acid or the TCA cycle, which eventually metabolize the intermediate to carbon dioxide and water, which is basically the end product along with the energy production. So कोई भी बायोमोलिक्यूल हो चाहे अमाइनो एसिड्स हों फैटी एसिड हों उनकी अमाइनो एसिड अगर डिग्रेड हो जाएं फैटी एसिड डिग्रेड हो जाएं जो मेन एनर्जी सोर्स यानी कार्बोहाइड्रेट हैं फॉर एग्जांपल ग्लूकोज है जो कि एक एक स्ट्रेट फारवर्ड एक मेटाबॉलिक पाथवे है जो ग्लूकोज इनिशियली ब्रेक डाउन होते हैं एज ए रिजल्ट ऑफ द ग्लाइकोलाइसिस और पायरुवेट बनाते हैं. They ultimately join the citric acid while making the acetyl-CoA. So acetyl-CoA you can say is one of the precursor molecule of the citric acid. लेकिन इसके अलावा भी बहुत से ऐसे junctions होते हैं जहाँ पे different metabolite intermediate of the different pathway they join the citric acid cycle और ultimately ये energy produce करते हैं. So either they directly produce the ATP या ये produce करते हैं some energy rich compound reduced form में for example NADH या FADH जो ultimately oxidize होके energy ATP equivalent energy produce करते हैं. So citric acid cycle TCA cycle का एक central role है energy production. It's a general overview of the TCA or the citric acid cycle, which always starts with the precursor molecule, which what we call acetyl-CoA. So acetyl-CoA basically oxaloacetate के साथ मिलके जो first product बनाता है वो citrate होता है. Citrate isocitrate में convert हो जाती है इसका isomer होता है जिसमें just rearrangement होती है कुछ internal rearrangement होती है कुछ functional groups की या सब्सिट्यूएंट्स की. फिर आइसोसिट्रेट कन्वर्ट होते हैं अल्फा कीटोग्लूटारेट में. अल्फा कीटोग्लूटारेट कन्वर्ट होते हैं सक्सिनाइल-CoA में. सो आइसोसिट्रेट में से कार्बन डाइऑक्साइड जब रिमूव हो जाता है तो अल्फा कीटोग्लूटारेट बनते हैं और साथ में ही NADH एनर्जी रिच कंपाउंड बनता है. और फिर जब अल्फा कीटोग्लूटारेट सक्सिनाइल-CoA में कन्वर्ट होता है तब भी एक कार्बन डाइऑक्साइड रिमूव होता है. तो basically जब कार्बन डाइऑक्साइड रिमूव होता है इट मीन्स द रिमूवल ऑफ अ सिंगल कार्बन. सो आइसोसिट्रेट से सक्सिनाइल-CoA तक दो कार्बन के मॉलिक्यूल्स हैं वो रिमूव हो जाते हैं इससे. और जब अल्फा कीटोग्लूटारेट से सक्सिनाइल-CoA बनता है तब भी एक एनर्जी रिच कंपाउंड बनता है NADH. देन सक्सिनाइल-CoA से कोएंजाइम A रिमूव हो जाता है और सक्सिनेट बच जाता है सिर्फ. और इस दौरान भी एक GTP का मॉलिक्यूल बनता है विच इज़ इक्वल टू अ सिंगल ATP. NAD सिंगल NAD जो वो 3 ATP अप्रॉक्सीमेटली 3 ATP इसके इक्वलेंट होता है. और फिर इसके बाद सक्सिनेट फ्यूमरेट में कन्वर्ट होता है. यहाँ पे एक FADH का मॉलिक्यूल बनता है. ये भी एनर्जी रिच कंपाउंड होते हैं जिनकी एनर्जी अप्रॉक्सीमेटली 2 ATP की इक्वलेंट होती है. देन फ्यूमरेट से मेलेट और फिर मेलेट से फिर ऑक्सैलोएसीटेट बनता है. और जब ये मेलेट ऑक्सैलोएसीटेट में कन्वर्ट होते हैं तब भी एक NADH रिड्यूस फॉर्म में एक एनर्जी रिच कंपाउंड बनता है जिसकी एनर्जी 3 ATP की इक्वलेंट होती है. अगेन ये बनने वाला जो ऑक्सैलोएसीटेट है ये एक अपकमिंग न्यू एसिटाइल-CoA से कंडेंस होकर सिट्रेट बनाता है एंड देन दिस साइकिल इट्स गोइंग ऑन. सो एक सिंगल एसिटाइल-CoA जो है ये 12 ATP के equivalent energy produce करता है. So this one is the more elaborated and explained overview of the citric acid cycle, TCA cycle, which is obviously starts with a precursor molecule acetyl-CoA, which condenses initially with the oxalic acetate, and this reaction is mediated by citrate synthase. Citrate synthase citrate का बनाने वाला. So when this citrate is synthesized, citrate is converted to cis econitrate, and this reaction is mediated by the econitase enzyme, and during this reaction dehydration occurs, means one water molecule is removed. Then this cis econitrate, it converts to isocitrate, and the reaction is mediated by the econitase, the one which breaks down the econitrate and makes the isocitrate. So this one is also a dehydration reaction because one water molecule is this one is the hydration reaction because one water molecule is added. So then this isocitrate is converted to alpha ketoglutarate, and this reaction is basically an oxidative decarboxylation, means oxidation, and the decarboxylation means removal of the carbon dioxide molecule, which removes a one carbon molecule from isocitrate. And this isocitrate is then converted to alpha ketoglutarate. The enzyme which mediates this reaction is isocitrate dehydrogenase. Uh, during this reaction, one NADH molecule is also synthesized. You can see that there are three reactions during which it leads to the synthesis of the NADH molecule. The one is the one during conversion of the isocitrate to alpha ketoglutarate. The other one is when the alpha ketoglutarate is converted to the succinyl-CoA, which is also a decarboxylation reaction during which one carbon dioxide molecule is removed and NADH molecule is synthesized. So then again, and this reaction is mediated by the alpha ketoglutarate dehydrogenase complex. Then the succinyl-CoA is converted to succinate, during which one GTP molecule is synthesized, which by means of the energy is equal to the ATP. This reaction is mediated by the succinyl-CoA synthetase. This reaction is also called as the substrate level phosphorylation because one of the phosphate is removed from the substrate. For example, here GDP acts as a GDP acts as a substrate, and an inorganic phosphate is added to this. So make a GTP, which by means of the energy is equal to ATP. Then this succinate and the acetyl CoA and the CoA is basically removed, which was added during the alpha ketoglutarate conversion to succinyl CoA. So then the succinate converts to fumarate, and this one is the dehydrogenation reaction. Dehydrogenation means one removal of the one hydrogen atom. So, and during this, another energy rich compound FADH is formed. Then this fumarate is converted to basically another intermediate which is called as the malate, and this one is the hydration reaction, and this reaction is mediated by the fumarase. Then this fumarate is converted to malate is converted to the oxalic acetate, and this reaction is mediated by the malate dehydrogenase, and then this oxalic acetate is, and during this NADH molecule is also synthesized, and then again the oxalic acetate, it merges or condenses with the acetyl-CoA to make the citrate, and so on. The reaction is continued in a cyclic manner. So this one is energy chart, which explains that how much energy is produced during a Krebs cycle when one acetyl-CoA it passes to the different intermediates. So during the single Krebs cycle, when one acetyl-CoA is converted to the different intermediate, produces 3 NAD, 1 FAD, and 1 GTP. So the energy equivalency of the NAD is equal to 3 ATP molecule. Energy equivalency of the FAD is equal to 2 ATP molecule. So as there are 3 NADH molecules formed during one Krebs cycle, so it means that a total energy equivalency of 9 ATP, and one FAD molecule is produced which equal to 2 ATP, and one GTP molecule is formed which by means of energy equivalency is equal to 1 ATP. So it means that one acetyl-CoA produces 12 ATP molecule when it undergoes through a Krebs cycle. So if we look at a single glucose molecule, uh, it means that a single glucose molecule, once it enters the acetyl Krebs cycle via the acetyl CoA, it produces 24 ATP only in the Krebs cycle, but during the glycolysis it makes some additional molecules. So if we look at a single glucose molecule, it roughly can make 36 to 38 ATP molecules. What's the role of the vitamins in the TCA cycle? As we have seen that there were different enzymes which were involved in mediating the different reactions, and each enzyme has for the sake of its activity depends on the coenzyme molecule as well. So all the coenzymes that are associated with the different enzymes are almost the vitamin B complex molecules. For example, riboflavin. So riboflavin, for example, in the form of the flavin adenine dinucleotide, which is basically a cofactor in the alpha ketoglutarate dehydrogenase complex and in the succinate dehydrogenase complex. Similarly, niacin, which in the form of the nicotinamide adenine dinucleotide, which is also a coenzyme for the 3-dehydrogenase enzymes. Thiamine, as a thiamine diphosphate, which is also a coenzyme for the decarboxylation in the alpha ketoglutarate dehydrogenase reaction. And then the pantothenic acid, which also act as a coenzyme. Coenzyme A, the cofactor attached to the active carboxylic acid residues, such as acetyl-CoA and succinyl-CoA. The disorder that are associated with the TCA cycles disruption, obviously if any of the enzyme which are taking part in the Krebs cycle, if they get distorted uh, or the deficiency of the enzyme occur, obviously it will produce a block, and the block means that the energy deficiency will occur, and obviously it will lead to some disease condition. So although it's the condition is very rare, because when the Krebs cycle is disrupted, this condition is non-viable. However, the viable very rare conditions include the pyruvate dehydrogenase deficiency, which is very rare, and it leads to developmental delay, intermittent ataxia, poor muscle tone, abnormal eye movement, etc. And the condition could be supplemented by limiting the carbohydrate and increasing the fat diet, and also can be supplemented with by taking the B complexes. The other deficiency enzyme may be the pyruvate carboxylase, which is also a very rare condition, and it leads to the poor feeding, vomiting, lethargy, mental, psychomotor, growth retardations. The other defective enzyme may involve the alpha ketoglutarate dehydrogenase, which is an autosomal recessive condition, and it leads to the psychomotor retardation, hypotonia, ataxia, convulsion, and different kind of other symptoms. Next defective enzyme may involve the succinate dehydrogenase, which is also an autosomal recessive condition, and there are 4 different genes that may be involved in the disease condition, because all these 4 enzymes product, they make a single enzyme complex, and they are the SDHA, B, C, and D. And the other deficiency enzyme may involve the fumarase enzyme, which is also a very rare condition, and it is an autosomal recessive amino the segregation, and its deficiency may lead to microcephaly, severe developmental delay, distinctive facial features, brain malformations, seizure, failure to thrive, and the hypotonia.